Good afternoon, everyone. It's a pleasure to be talking to this conference of emerging trends in basic and applied sciences. My name is Mohammad Haroon Qureshi. I'm currently working as assistant professor in Bosch University, Molecular Biology and Genetics Department. I joined Bosch University recently. Currently, for this presentation, I'm going to talk about one of the projects that I did for my postdoctoral work at Koch University. The title of the topic is Human Corneal Epithelial Cells as a Model for SARS-CoV-2 Infection of Ocular Compartments. We have been through COVID, and we remember all the trauma, all limitations. One of the things that is very prominent is that during COVID, it is the nasopharyngeal route that is the most common route for infection, and the mucus lining of your nasopharyngeal pathway and then your lungs, they are the ones that get most commonly infected. But what is important is that eye is actually another compartment which is very exposed to the outside world, and the anatomy of the eye makes it actually this mucous membrane right at the interface of the environment. So could it be possible that eye could also be, being a mucous membrane, could also be one of the route of infection? It turns out that this was the case, that a subgroup of patients that were getting infected with COVID were actually also reporting pink eye, which is the conjunctivitis. And this was reported in multiple ethnicities from multiple countries. For example, here it's an Italian cohort where it was shown that there is a conjunctivitis present among the COVID infection patients. There are other reports. For example, this published report actually said that there are ocular manifestations of COVID infection, most commonly as conjunctivitis. So what is conjunctivitis? It's the inflammation of the conjunctiva, which very often has presence in developing countries where there is a bacterial or viral cause of inflammation of our conjunctivitis. So how does COVID actually enter our cells? So it usually uses ACE2 as a receptor and TMPRSS2 as a co-receptor. Apart from ACE2 and TMPRSS2, there is also other receptors like NRP1 and furin, which could be used for virus to enter. Actually, there is evidence from other published work that ACE2 is expressed in conjunctivitis as well as cornea. And TMPRSS2, which is the co-receptor for virus to enter, is also present in our conjunctivitis as well as cornea. So now, okay, we have some evidence that people get conjunctivitis. We have evidence that cornea and conjunctiva may actually have the receptors and co-receptors for this virus to enter. But does it actually happen that eye could be one of the major major site of infection after of COVID. Here, this is the article which was published in Cell Host and Microbe, where entire mouse was actually studied. This is a humanized mouse, meaning it can be infected by COVID, and they wanted to see exactly what all tissues get infected get affected by the COVID infection. Uh, I looked into their data. They were kind enough to share their unpublished data with me. Now, what you can see here is this is the whole mouse, and this is the eye of the mouse. And inside the eye of the mouse, you can see the fluorescent presence here shown by spike protein, which is the viral protein. It's actually present after COVID infection around the eye, and this was a very good indicator. Here you can see it in the zoomed view. This was one of the positive signs that actually eye tissue could actually be infected by COVID. So this is the anatomy of the eye. The most outermost part, the cornea and conjunctiva, uh, is shown here. And if you look at the cornea, the outermost layer is actually consists of corneal epithelial cells. So what we did for this project, we took human corneal epithelial cells, which are immortalized. So this is a cell line, and we try to see whether in culture we can recapitulate COVID infection of corneal epithelial cells. Of course, there is some circumferential evidence that COVID can infect cornea and conjunctiva, but experimentally can we recapitulate that? So it turns out that yes, we can. So here what you are seeing compared to the negative control on the left, we have different concentrations, different MOIs, multiplicity of infection of COVID being incubated with corneal epithelial cells on top. So we can see fluorescent signal of spike protein expression in corneal epithelial cells after COVID infection. And the bottom, you can actually see that almost 72 hours after infection, the cells started to show cytopathic effects, meaning the cells started to die after COVID infection. So we planned to do an experiment where we would infect corneal epithelial cells with SARS-CoV-2, and followed by it, we will perform proteomics to see entire protein changes after COVID infection, and then perform functional clustering of these identified protein hits to ascertain what exactly are the affected functions and pathways inside these corneal epithelial cells. So this was our experimental plan. We would infect with SARS-CoV-2 in culture, and after 48 hours after COVID infection, we would collect the cells and perform their proteomics using LC-MS. So this is the relative abundance, showing that after 48 hours, cells have the overall abundance of the protein has not changed, meaning the cell hasn't undergone yet the cytopathic effect. Cell is still surviving, fighting for its survival, and this is a volcano plot showing the overall the proteins changes. So we identified 416 proteins that are misregulated, 228 are downregulated, and 188 are upregulated. Now, what does that mean? Out of these identified proteins, we performed something called protein network analysis, and out of all proteins that we identified, whether they fit into some smaller protein communities which have any functional meaning. So what you're seeing here on the top, there is there is basically uh, you have citric acid cycle and respiratory electron transport chain, metabolism. So these are the functional communities belonging to a subtype of protein network. So this is metabolism, citric acid cycle, respiratory electron transport chain, then slit and robo signaling, extracellular matrix organization, interferon signaling, and processing of pre-mRNAs as well as mRNA splicing. What these colors indicate is where exactly these proteins are localized. For example, extracellular compartment, cytosolic compartment, mitochondria, nucleus, or miscellaneous. So it is very well known that after COVID infection, metabolic reprogramming takes place. And this is indicated by previous studies where it was even recommended that could glycolysis inhibition in cells that get infected with COVID could actually be used as a therapeutic target. Slit and robo is peculiar because cornea is actually one of the most innervated tissue of the body. So per surface area, average surface area, cornea has one of the highest presence of nerves. And what slit and robo is indicate, basically slit and robo together, this pathway is meant for neurons, especially axons, to reach their target tissue, or axon guidance to be specific. So what happens is, so this article, another published work, it explored whether few months later after COVID infection, is there any neuropathy in cornea, meaning patients who have survived, who who didn't have any conjunctivitis or corneal infection after COVID, is there any presence of neuropathy in their nerves? And it turns out that a lot of these nerves showed effects which are matching neuroma, axonal beads, or dendritic cells presence. These are all matching some symptoms of dry eye disease and diabetic neuropathy, and there were morphological changes present in cornea even 10 months after they have recovered. So yes, there is a potential for slit and robo dysregulation and for corneal nerves to actually have been affected by COVID. Extracellular matrix organization is one of the most important aspect of cornea. You see in this figure, there are parallel organizations of collagen fibrils which are present in cornea, and any effect on this organization can lead to opaqueness of the cornea, which can affect the visibility and transparency of your corneal tissue. So it turns out that that there is a high probability of these pathways to get affected after COVID infection, as predicted by our studies. The next and one of the most important one is nonsense mediated decay pathways. So when RNAs are generated by transcription, they undergo processing to become mature mRNAs before getting transported outside. And nonsense mediated decay is one of the mechanisms that can degrade the mRNAs that have any premature termination codon specifically the stop codons. So virus genome and virus transcriptome is actually a multi, what you call basically many many genes get single transcript in form of mRNA. That means they have overlapping or contiguous presence of multiple mRNAs coding for multiple proteins. So this virus transcripts are a very good substrate for nonsense mediated decay that can target and degrade it. It turns out that after infection, the COVID, what it does, the first thing that it does is it basically it tries to limit nonsense mediated decay by inhibiting its core components through some of the viral proteins like NSP16, which binds to the UPF1, UPF2 proteins. And in our communities, nonsense mediated decay was one of the prominent pathways that was dysregulated. So a very strong indication that a lot of functions can get disrupted. By the way, nonsense mediated decay recently has been shown that there are almost 20,000 unique mRNAs that are targets of nonsense mediated decay, meaning there is a very wide spectrum of effects that can result after COVID infection, and a lot of pathways can get potentially inhibited. So we compared our results with one of the very famous studies here done by Boshkova et al. So what did they do? They infected SARS-CoV-2 cells, which are colon cancer cells, with SARS-CoV-2, and they performed a time-dependent analysis to study how what is the time-dependent changes in the proteome of the cells, and in effect highlighting what pathways and proteins get mostly disrupted after COVID infection. And their top hits included processing of pre-mRNA, TCA cycle and respiratory transport, signaling by roboreceptors, interleukin interferon pathways, TCA cycle. So this very closely matches to what we actually also found in corneal epithelial. So we wanted to see exactly at what time point in Boshkova et al. Our proteins match highest with their dysregulated proteome, and it turns out that our experimental data, it matches very closely with 24 hours after COVID infection in Boshkova et al. In colon cancer cells, KECO2 cell lines. So the overlapping proteins that we found in the overlapping proteins that we found between our studies and that of Boshkova et al., we use another platform called Omics Integrator, and we predicted certain clusters which get affected. So these clusters, again, not surprisingly, involved extracellular matrix components, peptide chain elongation, slit and robo, oxidative stress responses, and surprisingly, the fifth cluster about cardiomyopathy. And as you may remember, cardiac myopathies, the associated heart associated problems that emerged after COVID infection in certain cohorts studied around the world. So we didn't just uh, looked at the whole proteome of the cells, but we also looked at the secreted proteome. And we found that after COVID infection, the secreted proteins from these cells also show dysregulations. About 7 proteins were upregulated and 19 showed downregulation. These proteins mainly clustered in amyloid fibril formation, those affecting amyloid fibril formation, those involved in regulation of IGF receptors and extracellular matrix organization. So we compared uh, our proteins, for example, whatever happens in cornea, we were interested to see which tissue in the body closely matches uh, our altered proteome of corneal epithelial cells. So here in this article, what they looked at multiple tissues like lung, spleen, liver, heart, renal cortex, renal medulla, and testes from the patients that actually passed away after COVID infection, and they compared the they compared the changes in these multiple organs. We compared our proteome with these subsets, and we wanted to see closely which organ has the most overlap with our proteome, and our proteome, which is coming from cornea, closely matches uh, those coming from lung, liver, and spleen. Lung, liver, and spleen showed highest overlap. So this is actually showing the heat map among all these 7 tissues with that of cornea from our subsets. Okay, to summarize, we have shown that cornea get infected. Corneal epithelial cells actually show dysregulated proteome through protein interaction networks. We have shown and compared our findings with those reported by Boshkova et al. In experimental studies on colon cancer cells. We also shown the secreted proteome, and we also compared our studies with the multi-organ landscape. And to conclude, we have shown corneal epithelial cells in culture are susceptible to COVID infection. These changes induced post-infection are similar to those reported in KECO2 cells, which are the colon cancer cells. Liver is lung and spleen. These matches very closely to the corneal dysregulated proteome, and corneal tissue may experience lasting effects on the tissue structure, tissue architecture due to COVID-mediated proteome changes. So I acknowledge Koch University Translational Medicine Center, also known as QUITAM, my research advisor Afsun Shahin, and others associated with the studies like Murad Hassan Raisolu, Dr. Emil Sokullu, Dr. Fosunjan, Dr. Gulen Esken, and Typhoon Barlas. Our proteomics studies was done in collaboration with Kocaeli University, Dr. Murad Kasab, Dr. Mehmet Sarihan, Dr. Ali Yut Sevin from Istanbul Technical University helped us in computational analysis. And Dr. Ali Atuk from Helmholtz Institute of Tissue Engineering and Regeneration from Munich, he has shared kindly his research. So about me, As I said before, this is our university, Boss University. As you can see, it's set at the scenic Bosphorus Strait, and it's one of the most beautiful campus on the planet, and this is where we are located. My just quickly, my research focuses on RNA quality control mechanisms and their role in cell division and migration. So there is a very wide scope of for identifying the roles of RNA quality control mechanisms and how they affect cell division and cell migration. Both of these processes are important for cancer physiology. Just a quick introduction to some of my colleagues who are also interested in students from India, from Maharashtra, from Kolhapur. We are actively recruiting for PhDs and for postdoc applications. So Dr. Anas Kotil, he's working in mathematical modeling of biological processes, also interested in antibiotic resistance in bacteria. Dr. Ibrahim Charekurt, he is working on novel CRISPR proteins and variants. He has finished his PhD from Harvard University and has patents on novel CRISPR proteins. Dr. Erhan Kelesh, he graduated from EPFL Switzerland, did work in multiple universities, including Stanford, and his work focuses on computational experimental approaches in novel drug design. He also holds multiple patents in novel drugs. Lastly, Dr. Emine Kasanj, sorry about the panel not being visible, but she's working on metabolism of cancer cells, 3D organoids, cancer-derived organoids, and precision medicine. If you guys are interested in approaching us for PhD positions or for postdoctoral positions or even for short-term visits, please shoot us emails and we would be very happy to interact with you. So with this, I finish my presentation and I thank you all to listen to my presentation, and good luck with the whole conference. Thank you very much.